DNA fingerprinting is a technique used to identify individuals by analyzing their DNA. There are various methods for DNA fingerprinting. One of them is restriction fragment length polymorphism analysis, or RFLP analysis. Now let's see how RFLP analysis is performed. First, DNA samples from individuals are collected. Blood, semen, and hairs are the common sources of DNA. Then, DNA is extracted from the samples and cut with restriction enzymes. DNA fragments are obtained. Some of them contain variable number tandem repeats, or VNTRs, and some do not. What are VNTRs? VNTRs are present at many different loci in the non-coding regions on human chromosomes. Each VNTR consists of repetitive base sequences positioned one after another. At a given locus, the number of repeats varies greatly from person to person, and thus the length of the VNTR. Such variations in the lengths of VNTRs between individuals form the basis of RFLP analysis. As some of the DNA fragments produced contain VNTRs of different lengths, they can be separated according to their lengths by gel electrophoresis. Through special treatments, DNA fragments are denatured into single strands and transferred to a nylon membrane. The nylon membrane is then incubated with radioactive DNA probes. These DNA probes are short single-stranded DNA fragments that are complementary to the VNTRs of interest. During incubation, the DNA probes bind to the VNTRs of interest. Finally, the nylon membrane is placed on a photographic film. The radioactive probes expose the film, giving a pattern of bands. This pattern of bands is called a DNA fingerprint, which is unique to each individual.